Hello, everyone, and welcome to Chapter 6, Part 2, where we are going to focus primarily on population size as well as population growth. So let's go ahead and tuck on in to that. As a gentle reminder of how prokaryotes are going to reproduce, I wanted to throw in this slide about binary fission. So we already looked at this before in Unit 1, but let's look at it again very briefly. Remember that binary fission is going to be what we call cell division, but with bacteria, with prokaryotic cells. And also remember that this is a type of asexual reproduction, and the end goal is to have two cells that are going to be genetically identical to each other as well as to the parent cell. So that's starting cell. So there are just three steps to prokaryotic binary fission. That is elongation and chromosomal replication, septation, and then cell separation. So as a gentle reminder, that is how a prokaryotic cell is going to produce a new cell. Moving on to the rate of population growth, this is going to be how fast cells are able to produce new cells. Now remember from binary fission that we start with one cell, and once it goes through binary fission, we're going to get two cells. So we're going to have doubling of the cells. So we are going to increase the population by a factor of 2, hence doubling it, because each cell is going to be able to produce 2. So what we see is a doubling time, essentially, and this is going to be how we get a new generation. All right, so the generations of cells through time. Now, with the passing of each generation, again, that population will double over and over again. Now, you're kind of probably thinking that microbes would be overrunning us at this point, right? If they're able to just take a cell and then double it, and then those two cells double, and then those 4 cells double, and then so on and so forth. But there is kind of a factor to take into account, and that is that not all microbes are going to have a particularly fast generation time. So it is going to vary from species to species. So the average generation time is going to be roughly an hour or slightly less than an hour for a lot of microbes, but it can be even faster than that within 10 minutes. However, the flip side of that is that some microbes take months to produce new generations. So it just varies and depends on what type of microbe we're talking about, as well as what kind of environment that microbe is in. One thing I want to kind of present to you to think about is that pathogens have a really short generation time, meaning that they reproduce very quickly. I want you to think about for just a quick second why that might be. Our line of defense against microbes is going to be our immune system. So when we have these pathogens in our system that are causing issues in our body, causing an immune response, our immune system is going to kill those microbes, so they must reproduce quickly in order to outcompete our immune system. So even though our immune system is killing some of those cells, they are producing fast enough to supersede being killed off, which is kind of a neat aspect to think about. Also a little scary. So in this particular chapter, we are going to have a little bit of math that you're going to need to know, but we're going to lay it out like this first, and then I'm going to give you a bit of an equation to remember. So when it comes to calculating population growth, we need to think of the cell population being represented by the number 2, because remember that we are going to double those cells. So every time we are doubling the cells. So in the amount of time, however many hours or minutes or whatever, but we're probably looking at hours, we want to look at the number of cells and then have that exponent increase every generation. All right, so what we're going to end up seeing here is exponential growth. So this is going to be growth out of control. And we're going to look at a graph here in just a second that visually demonstrates what exponential growth looks like within a population, which can be a bad thing. So after some amount of time, let's say 10 hours, you have a ridiculous number of cells because they are just doubling every single time for every generation. The number that you see up at the top right of the 2 when it comes to the number of cells, so you've got like, I don't know, let's look at the half-hour slot. So you have a 2 and then in parentheses 2 and then a 1 up at the top there. So that 1 represents the number of generations. Where we get to the bottom at the 10-hour mark, we have a crazy big number and then a 2 and then 20. The 20 is indicating that it has been 20 generations. So I hope that makes sense. So here we have some visuals to look at. The first thing we're going to look at is this figure A, seeing that from 1 cell we get 2 cells, from those 2 cells we're going to get 4, from those 4 we're going to get, and it keeps doubling, right? So we are just doubling over and over and over, whereby the 5th generation you have 32 cells, and those 32 cells in the 6th generation are also going to double. So that is exponential growth. What that looks like on a graph is going to be figure C there at the bottom, where you have kind of the number gradually increasing until it hits a certain point and then just shoots right up, creating a J curve. Now, J curves are not sustainable, and that has everything to do with resources, but we'll talk about that in a bit. So here's that equation that I said that we would talk about. So make sure you have this equation highlighted, starred, something, and know what all the parts are to this equation. All right, so that first part is going to be the total number of cells in a population, where T represents a certain point in time. So potentially in hours is probably what we're going to be talking about. The next part is the starting number of cells. So however many cells we've begun with at that point. And then 2n is going to be the number of cells in that generation. So remember that the n part of the 2n is going to be the generation number. Could be anything between 1, 20, 100, a million, however many generations it has been. All right, guys, I am going to give you an example of what you might see on the exam. So our example here is if E. coli has a 20-minute generation time and there are 100 cells to start with, how many cells will there be in 3 hours? So what I want you to do is pause this slide, try and do this problem yourself using that equation that I gave you on the last slide, and then hit play when you're ready and see if you are correct. All right, let's check our work and make sure that we got this correct, and also understand what we're doing here, because sometimes it's easier to take an equation, actually plug in numbers, and see how that's going to go. So make sure that you have this equation written out somewhere close by this slide so that you know precisely what the equation is. Remember, it's in t equals ni times 2 to the n. So from the problem that we've been given, we can see that E. coli is going to produce a new generation every 20 minutes. So one of the first things you want to do is figure out how many generations there's going to be within the amount of time that we're asking. So we know that every 20 minutes, E. coli is going to produce a new generation, and we want to look at how many cells there might be in 3 hours. So if we let E. coli just grow for 3 hours, we need to figure out how many times 20 minutes is going to be within those 3 hours, because every 20 minutes we get a new generation of E. coli. So what you're going to do is take the 3 hours, figure out how many minutes are in 3 hours, which is 180. From there, take 180 minutes, divide that by 20. What you're going to find is you get the number 9. 9 is going to be the number of times that 20 minutes occurs in 3 hours. So there are 9 20-minute increments within 180 minutes. I hope that makes sense. So what you figure out is that n is going to be, that lowercase n, is going to be 9. That means we're going to see 9 generations within the 3 hours, because every 20 minutes, a new E. coli generation has been produced. We also can see from the problem that there are initially 100 cells to start with. So in I, that initial population number is going to be 100. From there, it's as easy as plugging and chugging. So once we have those numbers figured out, we are going to plug that in where we get 100 times 2 to the 9th degree, and then from there, 2 to the 9th is going to give us the number 512. Times that by 100, and we get 51,200 cells within 3 hours. I hope that makes sense. I would just listen to this slide over again if it did not. I can always give you another problem in class if you want a little bit more practice. More than happy to do that. So what you might be thinking after that word problem is that bacteria are going to kind of overrun the world, but that's not really going to happen, and here's why. In a closed system or a closed environment, there are limitations to resources that all living organisms need. So this includes microbes. When we look at these microbes, we know that they need food, they need space, and they can't tolerate a buildup of toxins, which is essentially like the waste product that the cells produce. So instead of seeing always an exponential growth curve, so growth out of control, what we more reasonably see is just a growth curve. So this is going to be a curve that ends up plateauing and then dipping down. So let's take a look at that. So this normal growth curve that we see with microbes is going to be much more reasonable, more realistic. So remember that this is going to be in a closed system, closed environment, where there is a limited amount of space, a limited amount of nutrients, as well as there's no way to get rid of the toxins and waste buildup. So we see this 4-part growth curve where we have the lag phase, which we'll talk about that in just a second, exponential growth, where the number of cells just shoot right up. So we get a very quick increase in population. We have a stationary phase where we're going to see the number of cells being produced matching the number of cells dying because we're starting to run out of those resources. And then we're going to see a decline in the population. So this is going to be indicative of the cells dying because there is no space, no food, or there is or also a buildup of those toxins that is killing off the population. So I want you to imagine with these 4 stages in a normal growth curve, that we are looking at bacteria being grown in a test tube in the lab. And what you're going to see in the lag phase is the bacteria getting used to or acclimating to its new environment, which is the test tube with whatever type of media. Maybe it's just a nutrient broth. Let's just imagine it's a nutrient broth. But what happens in the lag phase is these cells are not going to go through binary fission. They're not going to be dividing and doubling. That happens in the log or exponential phase. So that is where we see the bacteria dividing and the population is doubling every time the bacteria divides. And this is going to be a very quick growth of the population until we hit that next phase, which is the stationary phase. This is where bacteria are really intensely, like gladiator style, competing for resources that are now becoming very scarce. So this is including space, this is nutrients, and what's happening is that the cell growth is going to match the cell death. And what we see is kind of a stabilization of the population, which is why it's stationary. It's not growing, it's not shrinking, it is staying very consistent, it is stabilized. But that will not last for very long because we have a buildup of toxins and also a lack of nutrients, which is going to plunge us into that last phase, which is the death phase. So this is where bacteria are dying off. They are not dividing. So this is usually what happens in a media test tube or a petri dish in the lab, where we've grown the microbes for some amount of time, but we can only grow them for so long before those resources are depleted, and then the bacteria starts to die. So that's why it's hard to keep bacteria in the lab for a long period of time, and we have to grow it fresh for the lab every single time. The neat thing about understanding that microbial growth curve and those different phases is that we can kind of come to this neat conclusion that younger cells, you can kind of think of them like baby cells. They are not cells from a baby, but they're the newer cells that are produced from those cells reproducing, are going to be easier to kill or harm, or you can think of them as being more vulnerable. So what they're vulnerable to would be like antibiotics as well as heat or pressure. So they are easier to kill than the cells that have been around for a minute, kind of the more mature cells. So the newer cells are going to be easier to harm, especially if they're pathogens. So kind of an interesting thing to keep in mind. So here's another get you thinking kind of question. One bacterium with a generation time of 20 minutes, like E. coli, would, if it continued to grow exponentially uninhibited for 48 hours, would produce a population that weighed 4,000 times the weight of the Earth. So here's something to think about. Why haven't bacteria taken over the earth? And we've kind of answered this a few times, and that is because resources are limited. We are living on earth in a closed system. The resources it would take to support bacteria or microbes in general that have that type of generation time just doesn't exist. So what happens is they run out of resources, or competition with other microbes or other cells is going to limit that growth, as well as we kill them. So if you want to gently think back to COVID times, how we were constantly wiping everything down with Lysol, disinfectant, washing our hands, which you should still be doing, as well as hand sanitizer, things like that, there is this constant, again, I kind of am using the same word I think I used before, an arms race between bacteria and then other organisms, or microbes versus other organisms, as well as other microbes. So this constant pressure of limited resources, we're living in a closed system, competition, not only with other microbes, but also kind of competition to live in the form of organisms that constantly kill microbes, is what keeps microbes from taking over the planet within 48 hours, that kind of thing. So keep that in mind. Resources are limited. We're living in a closed system, competition, as well as organisms that kill microbes in general, like we do. So we have thus looked at being able to estimate the number of cells after however long based on the generation time of that species, which was super fun. I hope you enjoyed that in some capacity. Make sure you're able to do it for the exam. We are going to now look at population size and being able to analyze it, but not with math. So we can look at a culture, especially like a broth culture, and be able to estimate the population size based on turbidity. So this is just a real fancy science word for cloudiness. So if we were to look at a very clear broth, we know that there's probably little to no growth going on in there, versus that tube on the right, which is very cloudy. It has a high turbidity. That cloudiness is going to indicate that there are cells growing and using those nutrients inside of that broth. You can use different tools to estimate that size, but we can also just look at it with our peepers and know that the low turbidity tube is going to have little to no growth, while the high turbidity, we definitely have growth in that tube. Another way we can estimate population size or analyze the growth of bacteria is by counting. However, we do not do this anymore. We are not going to do this in lab, but it is a way that you can look at bacterial growth. However, it's not crazy accurate because you can't tell the dead from the living bacterial cells. There are automated versions that are more accurate in order to count these cells, but they're very expensive as opposed to just doing it with your own eyes. But again, you can't tell what's alive from dead. The last way to analyze population size of our microbes is going to be genetic probing. This is a type of PCR, which remember that is polymerase chain reaction. It can sometimes be called real-time PCR. What this is going to do is help quantify the microbes in an environment or sample without having to isolate them and without culturing them. Now, you might be like, what does that even mean? Don't worry, we are going to look at this later on in the semester in person, which is going to be super fun, super cool. So, hopefully you enjoyed this slightly shorter lecture over microbe growth. That was the end of chapter 6. So if you have any questions at all, please make sure to let me know. And remember, the vast majority of this chapter is definitions. So I would start studying up on this guy as soon as possible. Flashcards are going to be your best friend. And if you need other studying tips and tricks, let me know. I am happy to share that. Make good choices, and I'll see you for chapter 7.